This presentation will be on the genus Aedes. Here I have listed the 58 species of mosquitoes that historically have been found in Virginia. As you can see in the top left corner, there's a small little list for Aedes, and then in the center there is a large list under the genus Ocleritatis. Both of these will be talked about back in the late 90s, 2000s. Aedes encompassed all of these and then was split up into Aedes and Ocleritatis. Most recently, in the 2010s, it was reabsorbed back into Aedes. I have this list written this way just to give you an idea of the way that it was. So if you happen to see the term Ocleritatis in different ID books or in different journal articles, you understand what is happening. As you can see here, I have reorganized the list and put all of those species under the heading Aedes. From here on out in this presentation, I will be using the term Aedes and not Ocleritatis. But other presentations in this ID course, or when you go back to your jurisdictions, you might find that some people are still using the term Ocleritatis. It's hard to shake old habits, I get it. But Aedes is the more scientifically correct term to be using now. And as you can see, Aedes is the largest genus in the collection of the species found in Virginia. Largest as in has the most species under it. If this presentation were given at an in-person ID course, the species that are listed here in red are normally the specimens that we would be showing you, and you would be IDing them under the microscope. They're the more common of species found in Hampton Roads, and we're able to have collections to show you. Listed in blue are additional species that I'm going to talk about in my presentation, but usually we do not have enough specimens to show under the microscope. So how is the genus Aedes defined? I'm going to go over a couple characteristics that individually they may not be unique to this particular genus, but collectively these characteristics will define Aedes compared to all of the other genera that you find in Virginia. Some of these we may have gone over in other presentations, but I'm just going to bring them back so you know they're fresh in your mind. The first is the pointed abdomen. So at the top here I have a picture of a mosquito with a pointed abdomen, pretty prominently pointed. But below is a picture of a pointed abdomen as well. It might not seem as pointed because it's pretty much in comparison of what's pointed, what's rounded. Here I have a picture of a rounded abdomen. So when you look at the two, you can see how the one in the middle is more pointed than the one at the bottom. Also, sometimes the last segment of the abdomen, when the mosquito dies, that last segment will sometimes kind of pull inward into the abdomen. And so if that last segment were there, it might seem more pointed in the Aedes examples. And then at the bottom on the left, I have drawings of an Aedes, and I believe this is a culex, but pretty much showing you the pointed abdomen versus the rounded abdomen. The next characteristic is the banding on that abdomen. And the banding found on the specimens in the Aedes genus is going to have basal banding on their abdomen. And so hopefully we've already talked about, basal versus apical. If not, or if it's kind of forgotten a little bit, I'll go over slightly here. So the segment, when we're looking at the segments of the abdomen, we've got this line right here and this line right here. And this is one segment of the abdomen. Where the white band is considered the basal side of that segment. The other side, which is here, which is all dark, that is the apical side. And so one way to kind of remember, basal is closer to the body, basal body. Apical is away from the body, away, apical. Sometimes you might hear, people use the term, the apical is the ass end of the segments. And that works when you're talking about the abdomen. But if you're talking about the segments that are found on the head or the legs, it doesn't work as well. And so basically the body is considered the thorax. And so the apical end is away from the thorax. And so when you're talking about the apical end of the proboscis, you're going to be the very tip of the proboscis, where the basal portion will be closer to the head, closer to the thorax. And so sometimes this is the dorsal side of the abdomen, and sometimes the bands are very visible on this dorsal side. Sometimes the bands are more visible on the ventral side, the underneath, the belly, if you will. And then sometimes it is more visible on the lateral portion of the abdomen or the sides of the abdomen. As you can see, these segments have these triangles of banding, and they are all on the apical side of these segments. This last characteristic, the post-spiracular setae being present. So this is something that you really need to zoom in on and focus on. And a lot of the times this is not a necessary characteristic to look for because the other characteristics are enough. And you can figure out if it's point and end, if it has a point abdomen and it has basal banding, then you'll know that it's a Aedes. But what if the abdomen has been roughed up and you can't really figure it out from that? This last characteristic kind of gives you an idea if it's Aedes or not from this characteristic. And so what is post-spiracular? Well, all of these terms, post-spiracular, sub-spiracular, pre-spiracular, they're all based off of the spiracle. And so I have the pictures up top, or I should say on the right side, are two mosquitoes. You can see the spiracle here. This is a slit, and it's basically, this slit is a hole that allows air to flow in and out of. Mosquitoes do not have lungs, and so instead they have air come in, and then those tunnels go down, 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 smaller and smaller until each of them hit cells, and that is how air exchange is going to happen inside of the mosquito. Oxygen will go in, and carbon dioxide will go out. Here I have drawings of the side of the thorax, and you can see that the spiracle is here, the spiracle is here. Now, post-spiracular is behind, is after the spiracle. Pre-spiracular would be before, sub-spiracular would be underneath. And so the area that we're talking about in this post-spiracular is this area right here. So this like club-like shape is a certain plate that is found on the side of the thorax. You can't see it as well when you're actually looking at the pictures here, but if you have a nice microscope, you can see it. You can see here the shape that's coming down, but it'll be much more visible when you're able to actually mess with the microscope and look at it. But yes, this area right here, if there is setae, then that means the post-spiracular setae is present. If it is not, present, then it would not be an Aedes. Here I draw the outline of this plate, and then you can see the picture on the bottom does not have CD, where the one on the top there is CD present. And so the one on top is the Aedes, and then the below here, this is a Culex genus. There are several ways that I could divide out the species and create the order of this presentation. I could do it alphabetically. I could do it based on certain characteristics, but I'm going to divide it up by the larval habitat of these species. And so larval habitats kind of make some of these species associated with where they're found in the environment, and sometimes, you know, certain environmental conditions will make them come off at the same time. In this case, the first division is looking at species that are coming out of natural and artificial containers. So artificial containers are, you know, tires, trash cans, swimming pools, basically any container that is holding water for long enough. Even here, you know, gutters that have been backed up, or these downspouts that are coming off of the gutters. Natural habitats can be tree holes or rock pools. The first species we'll talk about is Aedes albopictus, also known as the Asian tiger mosquito. As I said before, this species lays its eggs and its larvae form in artificial containers and tree holes. This particular species is invasive into the United States. So around in 1989, this species first came in, and it has been spreading ever since. It has been spreading, you know, all along the East Coast and moving westwardly. There are, in the map down below, you can see it is spread more than this currently, but this was the most up-to-date map I found. As you can see, you know, there are some pockets on the East Coast, on the West Coast, and it is moving more and more as time goes on. For us in Hampton Roads, well, I guess depending on where you are, but I work in Suffolk, and this is the most common species for complaint calls. When citizens are calling about mosquito problems, this is usually what the problem is. They're, you know, coming out of artificial containers that are in your backyard, in your neighbor's backyard. They're biting you during the daytime, and so this is a very common nuisance for humans. They are bridge vectors for both West Nile and Triple E. They can also be a vector for Zika, dengue, and chikungunya. So if these diseases spread into the area, this species would be the primary vector of these viruses. The other vector would be a species we'll see later, but we do not really have much of them in Virginia. Northern Virginia has a little bit more. But they can be collected in large numbers from BG traps. If you are using CDCs with a BG lure, no light bulb, hanging low, you can also collect large numbers, or so I've been told, you can collect large numbers of albopictus that way if you do not have BG traps. And then populations usually peak in the summer. In Suffolk, we usually see a small peak in early May, and then that dies off or falls down, and then you see another peak probably around mid-June, and then it basically, that peak stays throughout the summer. Really high numbers in July, August, even into September. and the close-up of the map of the United States and how its distribution is. So what characteristics help define this mosquito? So they are black and white, as you can see in the picture. Lots of other species are, shades of brown and tans and copper. This one is a black and white mosquito. There are some other mosquitoes that are in black and white as well, but not an extreme amount. There is a thin longitudinal stripe down the dorsal side of the thorax, which I zoom into the picture, and you can see that white strip, that longitudinal line going down the thorax. The bands on the legs, they will be basal banding, so the segments of the legs, they will be on the basal portion of those segments, and there will be 5 of them. This is important later when we are discerning between another species, but you would see 1, 2, 3, 4, and 5. Each tarsal segment will have a basal band. And I zoom in here to show all 5 of those. And this is the hind leg. Sorry if I didn't say that earlier. A lot of these characteristics, usually in the key, in a dichotomous key, it will tell you if it's hind leg or mid leg, front leg, but usually a lot of the characteristics that we talk about are hind leg for mosquito species. The next species is Aedes aegypti, the yellow fever mosquito. This mosquito is not prevalent in Hampton Roads area. Every once in a while, you can find a specimen or maybe even a population of these, and then usually in the wintertime they kind of die off and they don't stick around. Currently around Washington, D.C., Northern Virginia, and portions of Maryland that surround D.C. are having a population of this mosquito that's actually probably been there for a while and it's sticking around. And there's, talk of where they're at, or they underground areas and things like this where the larvae is being found. But this species also is found in artificial containers, tree holes. It is also an invasive species into the Americas, but it is coming from Africa, and it's actually been around probably as long as Europeans came over to settle the Americas. So it's been here for a long time. It's found in southern U.S. states like Florida, Georgia, Louisiana, Texas, and there's a big pocket of them in Phoenix, Arizona, Southern California. There's population. So mostly southern, but they do come up, especially, human beings will bring containers when they move, and they were in their backyard and now they're putting them in their backyard in Hampton Roads. And so a lot of the times that will be why those populations pop up here and there, because the eggs have been brought up from the south. These mosquitoes are vectors for Zika, yellow fever, dengue, and chikungunya. These are actually better vectors for these diseases than albopictus, which we had talked about previously. And the reason being, better vectors for human beings, because these Aedes aegypti tend to feed on humans more often than albopictus. Albopictus is kind of an opportunistic feeder, feeding on lots of other mammals that are present, whereas aegypti tend to feed more on humans. And so these diseases are human-to-human diseases, so it's being taken from a human and then spread to more humans. They are also mostly daytime activity biters. Large numbers are collected in BG traps as well, and populations also peak in the summer. So lots of similarities with Aedes albopictus. Even the look of it, very similar. But when you compare the two, we'll be looking at the aegypti is on the left, the albopictus is on the right. Overall, the aegypti has a more kind of brownish look to it. So if you had a BG trap that you dumped out onto a petri dish and you're just counting away, looking at all these Aedes albopictus, and then all of a sudden there's just something that just stands out in that collection. You want to pick that out. You want to look at it. Maybe it's that overall brown color. Maybe it's the underside of the ventral side of the abdomen has this silvery look to it. So if something stands out like that, definitely take some time and look at it. Why did it stand out? And there could be a good reason for that. When looking at the markings on the thorax, the Aedes aegypti has this lyre shape of longitudinal stripes. And so you can see the lyre shape, which is found on the top of the thorax, opposed to the albopictus, which just has this one line, the longitudinal center. Silver scales on the clapius. So that is when looking at right above the mouth parts. So we've got, these are the eyes here. This is the beginning of the antenna here. Down at the bottom is the proboscis and the palpi. And so right above the proboscis at the very beginning is this clapius. And so you'll find silver scales on the aegypti. And so one way of telling the difference between aegypti and albopictus. Another one is looking at the mid leg of the aegypti. There will be a longitudinal stripe down the anterior of the mid femur. So when looking at, so we've got the femur, the tibia, and then you'll have the tarsi coming off to the right. And so when looking at that mid femur on the anterior side, you'll see this white stripe. And you'll not find this in books, in dichotomous keys, but I feel like the ventral side of the abdomen is very silvery when comparing it to an albopictus. A lot of the times this ventral side of the abdomen is really what stands out to my eyes, and I can pick them out. Or at least I used to be when we had a small population in Suffolk. The next mosquito is Aedes japonicus, also known as the Asian bush mosquito or the Asian rock pool mosquito. Larvae are found in artificial containers and rock pools. In Hampton Roads, we do not have many rock pools, so you'll find lots of these, most of these in artificial containers. But in the western portion of the U.S. or the western portion of Virginia, there are lots of rock pools, and in those niches you will find lots of the larvae of this species. They are also an invasive species into the U.S. They came in a little later, 1998, which is basically 22 years ago. It's not that much later, and they've been spreading throughout the U.S. since. They are more cold weather mosquito, so in higher altitudes and latitudes they can be more prevalent, and you'll find more higher populations of these mosquitoes proportionally in mountainous areas of Virginia. They are bridge vectors for West Nile virus, Triple E, and La Crosse. In Hampton Roads, populations usually peak in the spring and then in the fall. So you'll find them early on and then later on in the season. They can look very similar to Aedes albopictus, especially if you're just kind of glancing at a trap. But they're much larger than an albopictus, and sometimes that's one of the first things that pops out. Well, looking at the picture here, yeah, you can see them next to each other. You can also see some differences in coloration even by the picture. Obviously the markings as well. The thorax will have a golden longitudinal stripes. So very, looking kind of similar to a lyre shape, but not exactly. But instead of being black and white like the aegypti, it has this yellowish look to it. You can see the yellowish colorations. When comparing to the albopictus, when looking at pictures, it might be difficult to discern between the two. But when you're actually looking at the two mosquitoes, they definitely have their look. The hind tarsi has 3 bandings, and so this is one of the easiest ways to kind of determine which one is which, an albopictus or a japonicus. Again, an albopictus has 5 basal bandings on its hind legs, but a japonicus only has 3. Sometimes you'll find a 4th, but those are more rare. But you definitely will not find the 5. And I zoomed in here, and you see 1, 2, 3 basal bandings. And if you needed even more differentiation, the palpi scales are all black. So when you look at the palpi, the tips of the palpi will be all black. You will not see these silver ends of the palpi like you do in albopictus. White scales on the body have a bluish tint sometimes. So if you turn it just the right way, you can see instead of the white scales, it'll kind of have a metallic kind of bluish look to it. Aedes triseriatus is the next mosquito. This one is actually native to the U.S., or at least it's been around before human beings. It is also known as the eastern tree hole mosquito. Larvae found in artificial containers and tree holes. They are vectors for lacrosse, so they're the primary vector in the western side of Virginia, in the mountainous areas, that is going to be spreading lacrosse. They also can be a bridge vector for West Nile virus. Populations usually peak in the spring and fall, but you'll find them throughout the whole year, throughout the whole season. But I feel like they're currently in competition with albopictus, and so when albopictus numbers peak, then the triseriatus populations kind of fall. So they're being found more in the spring and the fall when albopictus numbers are lower. There's no banding on the legs of these guys. They're all black legs. They have large, brilliant white silver scales on the side of the thorax. And so when you see these in person under the microscope, they look really pretty. You can see these large, flat scales on the sides of the thorax that really, really stand out. The scutum, or the top of the thorax, will have a very large, dark medium area. And then there are lateral silver scales. So this is the top of the thorax, the scutum, and you see that this large dark area in the center, and then on the sides you'll see some white scales. The abdomen is laterally compressed, and so as we see this lateral view of the abdomen here, it's kind of pinched at the very last segments of the abdomen. It's kind of pinched laterally. You can see when looking from the dorsal side, these last segments are much thinner, but they're also much wider when you're looking laterally. They have incomplete dorsal banding. I mean, the banding is definitely basal, but it is not found on the dorsal side as much of the abdomen, but it is laterally, and the ventral side you can see that the basal scales are very prevalent. Aedes hendersoni. So this species looks very similar to Aedes triseriatus, and your biologists or the people you work with, they may not want you to differentiate between hendersoni and triseriatus. You're just going to ID them all as triseriatus, because sometimes it's very difficult to know the differences between the two, to see and understand the differences. But if your biologists are interested in looking at these, then great. You now know some of these kind of differences, or at least I'll show you in the next slide. Aedes hendersoni, they are also native to the U.S. They are mostly bird biters. You will find them, their larvae will be found in tree holes, but tree holes that are higher up in towards the canopy. So if you see a tree hole higher up in the tree and you were to dip it for larvae, presumably you may find more hendersoni than triseriatus. I have not had a lot of luck finding this larvae. The larvae itself is easier to identify, the difference between hendersoni and triseriatus. Very hard to differentiate from adult Aedes triseriatus, as I said before. Some of the differences: the lateral pale scales on the scutum have a more convex look instead of concave. And so when you're looking at these white scales, looking at this shape of the white scales on the scutum, they have this concave shape to them, whereas hendersoni goes more into the center and has a convex look to it. And then there are pale scales found on the base of the costal vein of the wing. So right there, kind of, if you think about it, like where your shoulder is, if you were to think of your body in comparison to a mosquito, if your arm was the wing, the shoulder area of that wing. So just right where it meets the thorax, you would find some white scales. I find it very difficult to differentiate the two, but it is fun to look and think that you have found one, and then you can pass it along to your biologist to see if you are correct. We'll now move on to the next group of species. So these species are going to be associated with woodland pool larval habitats. So mostly found in wooded areas, so areas where the water is very shaded. I get it, in these pictures they were all taken in the wintertime, so there's not a lot of shade going on. But not only what is above the water, but the water itself has a different chemistry when found in woodland areas, and that is going to also change the environment, the niche for these larvae. Aedes canadensis will be the first woodland pool species, and it is the woodland pool mosquito. You'll find them in woodland pools and woodland ditches. Their populations will peak in the spring. They are some of the first mosquitoes that are popping out in the springtime. Larvae can be found in late winter under ice. Possible reemergence in the fall. So you can find not only a peak in the population in the spring, but then a later population can be found in the fall, depending on environmental conditions. They are bridge vectors for triple E and West Nile virus. They are a pretty big nuisance biter. So when the populations are out, they're coming and they're biting all types of things, including humans. And if you cross their path, they're going to swarm you. Large distribution across the U.S. and Canada. One of the big determinations of canadensis is the hind leg banding, and it is going to be going across the joints. You can see that happening here, where we've got the joint right in the center, and the banding is on the apical side and the basal side. Again, the apical side and the basal side of these joints. So going across the joints. There are two subspecies found with canadensis, and they're both kind of differentiated by the width of these bands. And I believe canadensis has the wider, and then canadensis mathisoni has the thinner. But usually most of the Hampton Roads area does not differentiate between the two subspecies. The scutum, the top of the thorax, is going to be a solid brown. The abdomen is going to be mostly, the dorsal side of the abdomen is going to be mostly dark scales, as seen on the left here. You can see the lateral scales kind of popping through on the sides. The bands on the sides will be wide, and they will flow into almost an all pale scale ventral side of the abdomen. Aedes grossbecki, also known as grossbeck's speckled mosquito. These populations will peak in the spring. Larvae found in woodland pools and woodland ditches. I say that is uncommon in the Hampton Roads area, and that is true for certain cities. Certain cities have been finding them on the regular, like Hampton. For Suffolk, we have not found this species since the early 2000s. They are more common in the more northern and more western parts of Virginia. And then the distribution, mid-Atlantic and Mississippi River area. So the characteristics of Aedes grossbecki. On their wings, the scales are broad, truncate-shaped salt and pepper scales. So as you can see, the salt and pepper is this, some are black, some are white, interspersed. They are a broad shape to them, and they have this truncate look, which starts off thin, gets wider and wider, and then kind of ends. The hind tarsomeres will have broad basal banding. The picture might not look broad, but it is defined as broad banding. The scutum, or top of the thorax, will be dark brown median longitudinal area. So similar to the triseriatus, it has this dark brown top portion of the scutum. Prominent white lateral scales on the sides. And no banding on the proboscis. Aedes atlanticus. This is a woodland floodwater mosquito. Populations typically peak in summer and fall, and when they come out, sometimes they come out in large numbers. So after a large rain event, you can get 7 days or so after, you can get you can get large populations of these. Larvae are found in woodland pools, woodland ditches. They can be bridge vectors. They are bridge vectors for West Nile virus and Triple E. Aggressive biters. So if you walk into the woods where these populations are harboring, they will come and they will attack you. If you go running out of the woods and into an open field, it's like they kind of stop right there at the wood line. But they will come straight to your face and attack with purpose. Distribution in the Atlantic coast and southern states. So they are known for this white longitudinal stripe down the scutum. The stripe continues to the head to have this mohawk-looking area on the top of the head, and the stripe goes all the way down the entire scutum. And this is important when deciphering between some other species that might be rubbed. Legs and proboscis are unbanded. The abdomen banding is less, if nothing, on the dorsal side. And then there are these lateral triangles on the sides, on the ventrals, on the lateral portions of the abdomen. These triangles here. And then the ventral side is all white. Subsporacular area without scales. So before we talked about the post-sporacular area being after the spiracle. Pre is going to be before. Sub is going to be this area right here below the sporacular area on this like club shape plate. And so this is going to also be important if you wanted to use this characteristic to differentiate between this one and infirmatus, which we will learn about in a couple more slides. Aedes tormentor. This species looks very similar to Aedes atlanticus when you're looking at the morphological characteristics of the adult females. Not only that, morphologically, but they also both share very similar distributions across the U.S., very similar habitats and niches, and very similar behaviors. When you are looking at the larval stages of these two species, there are much bigger differences between them, and you can those characteristics have been known for a while, and you could differentiate them. But as adults, it's a little more difficult. And over the past decade and a half or so, new characteristics have come out to differentiate these two, and I'll go over these in just a second. But in this class, we're going to teach you the differences, but when you go back to your jurisdictions, maybe you will be asked to split these two, or maybe you won't. Sometimes it takes effort to split these two. You have to really zoom in on the head and take some time to know the difference between the two. And if we're just dealing operationally, control-wise, then maybe it's not worth it to split these two. But if you need to or want to, these are the characteristics. The biggest, most useful is looking at the head CD. So Aedes tormentor, when you are looking at the black lateral patches on the head, so on either side of that white mohawk, those black patches do not reach the eyes. So when we see, this is the tormentor, you have this white barrier between the two. It'll be 2 or 3 rows of narrow scales that will split the eye and this black patch, whereas the atlanticus will not. That black will go all the way to the eyes. This one is a little more difficult, but it's looking at the shape of that longitudinal stripe down the thorax. With tormentor, there is a slight pinch down here at the end, whereas the atlanticus goes straight across, does not have that pinch. Now, a lot of these specimens are going to be rubbed, and you're not going to see a very pristine stripe a lot of the times. And so that's why this one is a little more difficult to see, because I feel like even if this were a pristine specimen, you might think that, oh, is it really pinched, or am I looking at some rub happening and things like that. So so I go more with the head CD when I'm differentiating these two. Aedes infirmatus. So this one usually is differentiated wherever you are between this species and Atlanticus, even though there are some similarities. This is known as the silverback mosquito. Populations typically peak in the summer and fall after large rain events, just like Atlanticus. Larvae are found in woodland pools and woodland ditches. They can be West Nile virus or Triple E. vectors, bridge vectors. They can be aggressive biters. I feel like their behavior is very similar to atlanticus. You're out there in the woods. When you're in the woods, they can come and attack you. As soon as you run out of the woods, they kind of stop at that border. And then distribution in the U.S. very similar as well. But morphologically, there are some differences. So there are also some ecological differences. So in their niche, there is, after a large rain event, I forget exactly, but I think atlanticus pop off first. So after a large rain event, you'll see the larvae forming of atlanticus, and then a little bit later, you'll see the larvae forming for infirmatus popping off. I could have that in reverse, but there are some slight differences between these two species in aspects like that. But morphologically, that longitudinal stripe that is found on the thorax of an infirmatus is going to be much wider, and it's going to go down 2/3 of the scutum. So it won't go all the way down. It'll go 2/3 of the way down, and it'll be much wider. Also, the wide stripe kind of continues onto the head, and so having this mohawk that the atlanticus has, it's much wider on the head as well. And so if you get a specimen that's rubbed, and that is going to happen because the thorax is one of the first places to get rubbed, you might not see this very well. But if it is rubbed, you might be able to see that there's no line down at the bottom. And then the line could be the head, the CD on the head could still be there, and up at the top portion, the front portion of the scutum and the hind portion of the scutum. And so those rub less often. And so if you see that the scales don't go all the way down, but it is wide up at the top, then you could assume that this is an infirmatus. Again, there are no leg bandings or head or proboscis bandings, just like atlanticus. And the abdomen banding very similar as well as the atlanticus, where the dorsal side has no white scales, the ventral side has these triangles, and then all white on the lateral portion of the abdomen has the triangles, and then the ventral side of the abdomen will be all white. But one big characteristic that can help differentiate between these two is this subsporacular area. You really have to zoom in on your specimen to see this. So maybe it's worth it, maybe it's not. But if the thorax is rubbed and you just can't tell, you can scroll in and look at this subsporacular area. And if you find a patch of white scales right here on this plate, then you'll know that this is an infirmatus and not an atlanticus. And I use this quite often in the middle of the season. Once you get a hang of looking at it, it can become easy to use. Instead of flipping them over to see their thorax, sometimes it's easier. They usually lay on their sides, and it's easier to see this region. But, everyone has their own ways of doing things. I will now jump into the other larval habitats. So instead of woodland and artificial containers, natural containers, other habitats usually range in, open fields, so flood water, open ditches, marsh areas, areas with, cattails and permanent water, permanent pools, temporary pools, things like this, but usually open to the sun. Aedes vexans, the inland floodwater mosquito, larvae found in floodwater areas with sun exposure. Populations can be seen in the spring, summer, and fall. Kind of really depends on the rain events, but sometimes you can have a year with very few. Sometimes you can have a year with a lot. Bridge vectors for West Nile and Tripoli. and then the very wide distribution across North America. Aedes vexans will have hind tarsomeres with narrow basal banding. As you can see, the basal banding, the narrow basal banding here on the hind tarsomeres. There is no banding on the proboscis. This is the key characteristic usually used to differentiate this species from others. The basal banding on the abdomen will have this median notch, this W shape. So as you can see, the basal banding on the dorsal side of the abdomen, and it has this W shape with this notch. And yeah, so when seeing this particular shape, usually it was that species. Aedes cantator. This species can look very similar to Aedes vexans, and we'll go over some of the characteristics to differentiate. Brown salt marsh mosquito. So their larvae can live in brackish areas where brackish water kind of floods up onto the sides of the waterway, these little banks of the waterway. It can flood up and then remain there, and that's where these species can kind of be found. Population peaks in the spring. Bridge vectors for EEE. Distribution along the mid and northern Atlantic states, so not a very large distribution here, but we are just in the area. Actually, in this map we are not, but in Suffolk we definitely find these species in a couple trap sites. They share many morphological characteristics with the Aedes vexans. When you are first IDing, these species can look very similar to a vexans, and they can be mis-ID'd very easily. But if you're looking at a large cluster of vexans and there's a cantator in there, cantators can stand out pretty well color-wise. They're brownish instead of vexans are more black and white. And then the other characteristics that we'll go over here. There are no banding on the proboscis, and the hind tarsomeres have narrow basal banding, just like vexans. Overall, more brownish gold than vexans, and that color can really stand out if you know what you're looking for. The basal banding on the abdomen does not have that median notch. There's not that W shape. Instead, it goes fairly straight across. And the last two segments are going to be entirely pale. And then that is easy to see for a lot of specimens where vexans will not have that. And then the underside of the abdomen is really helpful. The ventral abdomen of cantator is going to be entirely pale, whereas Aedes vexans will have the dark portions of the banding will be seen on the underside. So you can see the vexans here, the dark side is showing through, going all the way around, whereas the cantator is going to be entirely pale. Aedes sollicitans, the eastern salt marsh mosquito. Larvae found in brackish waters on the edge of marsh, so very similar to the habitat I was explaining for cantator. Tidal floods, as the tidal flooding takes place and then dumps over into the bank, onto the bank, and pooling on the bank when the water recedes, those pools remain, and that's where these species can breed. Population usually peaks in the summer, but you can have some earlier or later species populations. Bridge vector for EEE, West Nile. Distribution along the coastlines of eastern U.S. and Gulf states. The proboscis will have banding on this specimen. And you can see this is I think the first specimen, the first species that we see with banding on the proboscis. And you can see here the banding on the proboscis, right in the middle. The hind tarsomeres with wide basal banding. So we see we've got a basal band here, a basal band here, a basal band here, basal band here. But this band is going to be a median pale band. It is in the middle of this segment. So the segment starts here and it ends here. This band is in the middle of that hind tarsomere 1. The wings are going to be speckled with dark and pale scales. And the thorax with white scales on the sides, as we see here, and yellow scales on the scutum. There is a median longitudinal stripe down the dorsal abdomen segments. And you might not find this in the key when you're using a dichotomous key, but this is used to differentiate between some other species, but you can see this longitudinal stripe going down the abdomen. So you see in this case we have basal banding. Basal banding. Here it looks like you've got basal and apical banding. So don't be confused there. The sides of the thorax will have there will be basal, and then this longitudinal stripe will be found in the middle. Aedes taeniorhynchus, the black salt marsh mosquito, also found in those brackish water edges. Populations peaking in the summer. Bridge vectors for Tripoli and West Nile, and a similar distribution in the U.S. to taeniorhynchus. To sollicitans. Sollicitans tend to be larger mosquitoes than taeniorhynchus. Taeniorhynchus tend to be a little smaller between the two, but they might come off in the same areas environmental-wise. Taeniorhynchus, the proboscis has a narrow band. This is the narrow band here. Hind tarsomeres with basal banding, but there is no large median pale band on hind tarsomere 1. So you see it has the basal banding on the tarsomeres, but there is no basal band here. And so if you're trying to determine between these two species, this is a big characteristic, looking at that hind leg and seeing that it's not there. If you were trying to differentiate this species from vexans, you would notice that it's going to come up later. The proboscis will have that. Oh, it was the first one. The proboscis having the narrow band. So looking at the difference between this and vexans, obviously you can look at the abdomen, but the proboscis band on taeniorhynchus and not on vexans. The wings will have all dark scales. They will not be speckled like sollicitans. The abdomen with basal bands lacking that median longitudinal stripe. And as you can see, the white scales make these like triangles on the sides, the lateral portion of the abdomen. It's kind of hard to see, but I tried to zoom in there. The thorax with white scales on the sides, and scutum is brown. I believe this might be the last one. This is Aedes fulvus pallens. They are very interesting looking mosquitoes. They are the eastern yellow and black mosquito. Larvae found in woodland pools, usually along the side of Aedes atlanticus. Populations peak in the summer. They do not transmit pathogens. They are large, golden orangish mosquitoes. At a quick glance, they might look like a mosquito that has all of its scales rubbed off. The first time that you see these, they can be very, very alarming. You think that you found something really weird. This mosquito has gone through some crazy life event. But nope, that is just how they look. Two large posterolateral black spots on the scutum, as you can see here. Two black spots looking shiny. And then the legs, proboscis, and palps all have are yellow with these black tips. It's kind of hard to see with the palp, but you can see the black tip on the proboscis. But you should be able to see it as well on the palps. And then on the legs, you'll see in this top picture, you can see the black kind of like banding on the legs. So that is all the specimens I have for you looking at characteristic wise. I did skip over some species that are a little less prevalent in our area, but you still could run into these species. So the list here to the left, 80 species found in Virginia but not discussed in the presentation. Even though these are rare, you could still find them in the Hampton Roads area. In fact, Suffolk, over the last 15 years, we have found all of them except for stimulants. All of these others we have found in the last, I have 10 years, but I think this presentation is a little older. We have not found grossbecki either, which is disappointing, but they did find it in early 2000s. It's good hunting. Good luck IDing and good luck hunting for these mosquitoes.